Now I would like to introduce Dr. Grant Schulert. He joined our division about 4 years ago, and we share a strong passion for studying macrophage activation syndrome and systemic juvenile idiopathic arthritis. Dr. Schulert.
Good morning, everybody. First, thanks to John and Rashmi for asking me to participate in all of this, but most importantly, thank you to all of you for making the trip here to Cincinnati and taking the time on a hot but otherwise nice Saturday morning to see what we can all do about this diseases together. So I was given a somewhat daunting charge, which was to discuss research updates in systemic JIA and MAS. Which is daunting because it's actually a very exciting time in the field. For a lot of reasons, there's a lot of interesting discoveries that have been made and are being made. In fact, there are whole day workshops on this topic that I've been to, which can be very confusing and overwhelming. So instead, I will endeavor to overwhelm and confuse you in only 20 minutes instead. So to try to sort of focus my thoughts, I tried to think about a couple of really key questions in the field that there's been some interesting studies that have been starting to examine. And these are questions I hear all the time from families and patients. And the first one really is, what is the cause of systemic JIA? And spoiler alert, I can't answer either of these questions, but I think that we have some interesting findings that have been made in the last, really in the last couple of years. So as has been discussed by particularly Dr. Bruner, systemic JIA is not the same as arthritis, as juvenile arthritis or rheumatoid arthritis. And those are really what we call these autoimmune diseases. And in autoimmune diseases, parts of your immune system, particularly things like T cells and antibody-making cells, attack tissues in your body. In diabetes, it attacks your pancreas, which makes insulin. In arthritis, it attacks the lining of your joints. But this is very different from systemic JIA, and systemic JIA is largely what we think of as this autoinflammatory disease, where rather than T cells and antibodies attacking tissues of your body, what happens is that parts of your immune system, particularly those first-line defenses, turn on, and it has these really seemingly unprovoked or random episodes of systemic inflammation, and then it sort of can't turn off. The model of these that had been first described were these genetic periodic fever syndromes, things like familial Mediterranean fever, but really much of systemic JIA sort of fits this model. So thinking about what are the causes of these complex diseases we take care of? You know, a lot of the models for how we think of this come from cancer, because we spend about 100 times researching cancer. What we spend every year researching cancer is what we research inflammatory arthritis. But if we use cancer as our model, on one end of the spectrum you have things that are almost exclusively genetic. An example, this familial cancer form, where people have a mutation in this disease and everybody gets colon cancer. It doesn't matter, you eat the healthiest diet, you get a colonoscopy every week, it doesn't matter, you're going to get colon cancer. On the other end of the spectrum, very severe environmental exposures, so like folks who worked at Chernobyl when it melted down. You can have the best genes in the world, you're going to get cancer because you worked at Chernobyl. Unfortunately, most complex diseases are somewhere in the middle, where we think there are components of things in the environment that contribute to this, but then there's gene and typically many genes that contribute to this as well. So as has been mentioned, systemic JIA is rarely familial. There are a few reports of siblings or twins that both have the same type of the disease. But there are hints that there are some genetic risk for this. Probably the strongest one is that there is different incidences in different ethnic groups, particularly in parts of Asia, such as Japan. Some Northern European populations have higher risk of this than Southern European, and that persists even once they move to, say, the United States. So we think this isn't just environment, this is probably genetic to some degree. So when we think then about our genes and sort of how that contributes to disease, each of us, when we look at our DNA, we're over 99.5% identical to everybody else on the planet, which sounds really, really close until you realize that that really means that each of us has probably 10 to 20 million differences in our DNA from anyone else. And so understanding which of those differences make no difference, which ones of them are the ones that just make me you and you, and which ones contribute to our risk for diseases is really challenging. Most of those 10 to 20 million changes are these single nucleotide changes. And we call them sometimes SNPs, or single nucleotide polymorphisms. So for example here, let's say at this position here, 94% of people in the population have a G or a guanine in that position, but 6% of them have a T or a thymine in that position. So we call that a SNP. And again, we have millions of these. And so understanding which one of these are important and which ones aren't important is the work of very smart people who have been studying these genetic causes for a long time. And one of the great tools for doing this is something that's been called these genome-wide association studies. And what these are studies where they look at tens of thousands or millions of these small changes between people and try to figure out which ones of these are associated with disease and which ones aren't. And so to do that, you have to look at hundreds or thousands of patients and compare them to hundreds of thousands of people who don't have the disease. And then what they do then is they line up sort of all of them in a row, just like we think of the genome as a row, and then look for which ones have sort of the strongest association with the disease. And that's called a Manhattan plot, because when you look at it, it actually looks like the skyline of Manhattan, where you see all of these this sort of low area around the thing, but then these peaks. And those peaks suggest areas or genes that might be important for a disease. So the work of this in non-systemic GIA has actually been led for a long time by folks here at Cincinnati. David Glass, who is the longtime director of our division here at Cincinnati, and now Sue Thompson, who is still here in the Center for Autoimmune Genomics as well as Rheumatology. And they've published extensively on this in what I call regular GIA or conventional GIA. And their study from a couple years back, which was really a landmark study in this field, looked at nearly 3,000 patients with other forms of GIA, oligo or polyarticular GIA, compared that to 13,000 controls. And they looked at many, tens of thousands of little changes to try to find areas they thought were important. And then this is that Manhattan plot, and you can see that here's Freedom Tower, right, the big one right in the middle. And you actually see there's sort of a break in this, so if we didn't have this, this one would really be off the chart here. And then other little buildings were popping up also as important, but this one was really the most striking thing. And what is that? That was what we actually called the HLA region, or the major histocompatibility complex type 2 region. And what this is in the immune system is the part of your immune system that actually presents peptide antigens or proteins to T cells and sort of shows them to T cells and says, is this self and something I shouldn't worry about, or is this a virus or an infection or something that we should get really excited and fight? And the MHC controls what things are presented and how they're presented to your immune system. And we think that in a lot of autoimmune diseases, this is what breaks down, right? That because of how they present them or the kinds they present them, some of these are more prone to having mistakes that causes autoimmunity. So that really makes sense, and really because we think of regular GIA as an autoimmune disease. And all those other smaller buildings, many of them were other genes that have been known to play roles in immunity or other autoimmune diseases. So just over the past year or so, a large-scale study of this has been published in Systemic JIA. This was led by Mike Umbrello at the NIH, but was really a large-scale collaboration between many groups around the world, including folks here at Cincinnati. And they looked at nearly 1,000 children with Systemic JIA compared to thousands of controls. And they really found sort of 9 geographically defined matched groups of patients. So this isn't just what do American GIA patients look like, it's also what a Brazilian GIA patients look like, what do Italian GIA, systemic GIA patients look like. And they looked at thousands and thousands of these single nucleotide changes. And here's what they found, and this looks less like a Manhattan plot and more like a Peoria plot, because you can see that the buildings here are a lot smaller. There's not that giant dramatic peak. There's no Freedom Tower here like we saw in the conventional GIA study. But interestingly, what they did find was the tallest one was in the same MHC region, which is interesting because the dogma was that, well, the T cells and that parts of the immune system don't really play a role in systemic GIA. It's all this innate immunity. And this suggests maybe that's not the case. This is much weaker of an association than they saw with regular GIA, but it's definitely real. And so they did find some areas in this that were associated with about a 2.5-fold increased chance of having systemic GIA. So if somebody has these changes here, their risk of developing the systemic GIA is 2 or 3-fold higher than somebody who doesn't have that, which isn't huge, but it's interesting, and it may tell us something about what's causing this disease. These other genes that they found were actually very, very interesting. So they found about 24 other genes that seemed like they may be playing a role as well. None of those genes had ever been associated with any other rheumatic diseases. And when they looked at the sort of the collection of these compared to what they found with other forms of JIA, completely different. So really confirming that this is not JIA, which all you guys know that already. You didn't need an expensive NIH study to tell you that. But now we've got genetic evidence of this, that this looks completely different than regular JIA. And the hope is that then by trying to understand what these genes are, we can understand better what's causing this disease, but even more interestingly, be able to predict who's going to be at risk for this type of disease. So now to change gears a little bit and talk some about MAS and kind of the key questions of what's happening during MAS and why do only some patients get this, right? So maybe 1 in 10 to 15% of children get this very severe MAS where they end up in the hospital or in the ICU, and as Dr. Grom said, maybe a third or more children can have this sort of smoldering and low-grade MAS. But still, that means a majority of patients with systemic JIA never get this, and why is that? So Dr. Grom sort of showed this here as this sort of mechanism of what we think is happening in MAS, where we get this systemic cytokine storm. And we think one of the key drivers of this is interferon gamma, as Dr. Grom mentioned. So interferon gamma in the HLH world has been studied for quite a while and is really felt to be one of the key drivers of this. They see high levels of it in patients. Mice that have these same genetic changes that children with genetic HLH have, if you block interferon gamma, those mice do just fine. And as Dr. Grom mentioned, there's also very promising results from a clinical trial of this. So the research question is interferon gamma doing the same thing in MAS as it is in HLH? So this was a paper that was published last year looking at children with systemic JIA, a large group of our patients here, as well as them from Genoa as well as Rome, and looking at some of their cytokine levels. And so it's very interesting. We looked at several groups of patients, children with systemic JIA who were doing awesome. They had no signs of disease at that time. Patients whose disease was active but they weren't having MAS. And patients who were having MAS, and as a comparison, also looking at patients with this HLH. And what we found was that, in fact, interferon gamma levels were much higher in active MAS than they were during systemic JIA without signs of MAS, even when the disease was very active. And even more so, these are actually three proteins that are really turned on by interferon gamma, so they're sort of a secondary measure of interferon gamma. And we would see, 5, 10, 20, 50-fold higher increased levels of these during MAS than we see during active disease without any signs of MAS. And then we had several patients where we were able to study them both during MAS and at times when they had a flare but no signs of MAS. And we would see that even within a single patient, then the levels of interferon gamma, but more importantly even these markers of interferon gamma activity were really excellent predictors of MAS. And those levels also then correlated with some of those laboratory features that Dr. Grom mentioned of MAS. So high levels of feridem in patients with full-blown MAS were very strongly associated with the levels of interferon gamma, and particularly this interferon-induced product. Dropping cell counts of neutrophils and platelets, as well as the signs of liver inflammation and liver damage. So this is a very exciting because it suggests, first off, this can help us diagnose MAS, but most importantly, this really supports that potentially blocking interferon gamma during acute MAS could be very helpful for these patients. Dr. Grom also mentioned IL18. IL18 has become a very interesting and exciting molecule in systemic JIA and MAS. It's actually something that really drives this interferon gamma production. Really, before this was linked to our diseases, this was understood in how your body fights infection. Macrophages recognize some kinds of bacteria. This is salmonella, which hopefully no one will get from our lunch coming up next. IL18 is produced by your immune cells sort of as an alarm signal when it sees some of these infections, which then turns on T cells, which then make interferon gamma and sort of help fight off this infection. And this was really demonstrated a couple years ago by one of our colleagues, Scott Canna, who's now at Pitt, who's a pediatric rheumatologist, who found a patient with a genetic recurrent MAS. They didn't have systemic JIA, but they kept having episodes of MAS, and found that he had a mutation that really produced IL18 all the time, and really linked that with recurrent macrophage activation syndrome. There's also been very interesting work on this by some of our colleagues in Japan, Masaki Shimzo, who really his data has shown that there's a subgroup of patients that have high IL-18 levels, and this really overlaps with the ones who get MAS. And particularly compared to IL-6, which we think is a big driver of arthritis, is if you kind of plot these against one another, patients who fall on this side of the fence and have this sort of IL-18 predominant are the ones who get MAS. And those green dots here, I don't know how well, that projects reasonably well. The green dots are the ones who have MAS. And all of them fell on this IL-18 side. None of them fell on this low or lower IL-18 side. There's a fellow in our lab who's working on replicating this in some of a US-based cohort, and we're seeing similar results, that it seems like higher IL-18 levels are really associated with the patients who get MAS. And then finally, Dr. Grom had talked some about sort of potentially the genetic relationship between MAS and this genetic disease called HLH, which has been linked to a number of different genes that control how the movement of all of these cytotoxic granules. And all of these things here, mutations in all of these have been linked to these genetic forms of HLH, which is very similar to MAS. So over the last several years, multiple papers had reported sort of single or a couple patients who have systemic JIA, have recurrent MAS, and had mutations in some of these genes. So the question that we had asked is, could this be in fact a genetic predisposition to MAS? Could this explain why some patients get MAS and others don't? So we, Dr. Grom and others here, looked at what we call a trio-based exome sequencing, where we sequence both a patient as well as their two parents, with patients who had systemic JIA and recurrent episodes of MAS, as well as looking at patients who have systemic JIA but no MAS. And looking first for variants in some of these genes that are associated with MAS, but also then looking for other things, right? Are there other unknown genes that could be predisposing these children to MAS? And what they found was very interesting. First, as Dr. Grom said, more than a third of these patients had known variants that have been associated with HLH. And this has been similar to a couple other studies that have found a similar number of about a third of patients who have MAS have these variants, which is significantly higher than we see in patients who have systemic JIA but no MAS. But interestingly, when you do this sort of whole, I don't know if several of you, I know your children have had whole exome and whole genome sequencing. You get lots of information. You don't necessarily know what to do with it. But then sometimes by then looking at a group of patients and kind of lumping the all together, you can kind of try to see some patterns. And when we did that with this group of, I believe, 14 patients, we actually did see a pattern, which was that there seemed to be, an overrepresentation, of genes in this pathway that's been called cellular organization and assembly. So they seem to be more likely to have rare changes in genes involved in sort of movement of these granules within cells. So it suggested that some of these other genes that we find could in fact be genetic risk factors for MAS. So, putting this kind of together to see kind of what is our current model for what's happening during MAS, I think there's very strong evidence that at least in some patients there's a genetic predisposition. Around a third of patients who have MAS, and particularly multiple episodes of MAS, we can identify genetic changes that look like what we see in HLH. So that's part of it. And then there's this inflammatory background, this cytokine storm, where these high levels of these cytokines that we've talked about really turn on the immune system and cause that to really malfunction. And particularly IL-6 seems to be something that really drives that, as well as IL-18. And then often there's a trigger. As Dr. Grom mentioned, what we often see is patients who are doing awesome on one of these treatments, then they get a respiratory virus or some sort of a febrile illness, and then it kind of tips them over into MAS. So that we have an immune system that's kind of delicately balanced, and then an infection that kind of tips it over, and we end up getting this cytokine storm. And we think that interferon gamma is really a key driver of that, and probably a very promising target for treatment. Now, like any good scientist, anytime you answer a question, you get 2 or 3 really good ones that come out of that. And I think the questions that we're asking going forward in the next several years from this data is first, as we're understanding more about what are the genetic causes of systemic JIA, how do those genetic factors as well as environmental triggers interact to really cause systemic JIA? Secondly, as we identify more of these cytokines that we think are key in MAS, can cytokine-directed therapy, particularly against interferon gamma or IL18, can that be used to treat MAS? And most sort of interestingly, can genetic testing or cytokine testing really predict the risk for MAS? Because I think it would be really nice to know which patients we need to be worried about MAS and which patients we think are at a low risk for MAS. So I believe that's my last slide, yes. I would be happy to take any questions. I know several people had mentioned things earlier that then got kind of punted to Grant and Alexia will talk about that. And if we haven't talked about that, I've sort of scribbled down a few notes here, which we can talk about as well.
Oh, were you gonna? Oh no, okay. Can you just clarify? Still's disease is the term used for adults, and we're in a unique situation because our daughter is now 19, and her SJIA is now called Still's disease. I know in our group someone posted an article that there's more information saying that SJIA has the same gene expression as Still's disease, but we're not really bridging these two worlds together.
Right.
Can you just clarify that for us? I mean, is that accurate, that SJIA and Still's disease is the same disease?
I think that we think they're the same disease. And as we understand more about genetics, I think we'll be able to prove that. But I think that the paper you mentioned actually is probably the strongest evidence for it, is when we look very closely at what the immune system looks like, it looks very, very similar between systemic JIA and Still's. And in fact, we can ask Dr. Lovell about that next. There's a sort of an international movement to sort of rethink how we classify arthritis and what we call it. And some people want to actually go back to calling systemic JIA Still's disease because it really is different, right? And by calling it systemic JIA, it's like, oh yeah, it's just like, the kid whose knee is swollen, that's JIA too. And we're like, well, it's not really the same thing. So there's at least some thought to go back to calling it Still's disease, which is kind of funny, because that's where we were 20 years ago, 30 years ago.
I'm sure this is a question that you'll only be able to answer in generalities, but my daughter's fairly new to being diagnosed with this within the last year. Is there a point where, beyond going through all of these types of testing, which I have no clue how to do, we haven't done it, but is there a point where you can say, okay, if they haven't manifested having MAS within the first year, you're probably okay? Or is it just one of those things, hey, worry about it for the next 20 years, good luck?
Right.
Okay, that's my question.
That's a great question. I would say that most patients, but certainly not all patients, most patients who have MAS have at least features of that at the beginning and at diagnosis or earlier in the disease. There are absolutely patients, and I think at least one of the parents in the panel had said that they never had that until a couple years into treatment. And I have patients like that as well. But I think most patients who have MAS have those features at the beginning.